Salt stress is an issue. 30% of all irrigated land in the US is affected by salt stress, and this is a problem that worsens every year. Plants that endure salt stress experience reduced yield, premature flowering, and even premature death. So clearly this is an issue that we need to solve. And Brassicaceae is a great family to solve this issue in because there are many members that are salt tolerant, but also has close relatives that are salt sensitive. For example, Arabidopsis is a model system. It's genetically tractable. And by genetically tractable, I just mean that it's easy to work with in the lab and it's transformable. However, as you can see, it's salt sensitive. Conversely, Camelina sativa is salt tolerant. It's also genetically tractable. And Eutrema is also salt tolerant and genetically tractable. So I'm thinking if we can investigate how these salt tolerant species protect themselves from salt stressors, we may be able to ask some good comparative questions to help us solve this problem of salt stress. So these are the species I just mentioned, and to salt stress them, we incrementally applied salt by 50 millimolars until it reached its max concentration, and then applied an intense systemic salt stress for 2 weeks to get our samples prepared. And then they were sent for RNA sequencing. This is the overall project workflow. So the samples, as I mentioned, RNA was extracted and sent for sequencing. And then I come in to do the bioinformatic analyses. And I'm very hard at work taking selfies in the office. So that is the office I work in. And when presented with RNA sequencing data, the first step is obviously to clean and filter it so it's quality. But after that, we wanted to identify which genes were changing as a result of the salt treatment. So these are the representative heat maps for that. Warmer colors like pink and reds are going to represent higher expression. And as you can see, the control and salt samples are expressing different genes. However, I wanted to do another quality check just to make sure that the transcriptome was acting how we might expect under stress. That's what the gene ontology term plot is for. So on the y-axis here are our terms. On the x-axis is fold enrichment, which just means how many times greater a gene was seen than compared to what it was expected. There's also a color scale, so reds are going to represent more significance. However, they're all very significant. And then the size of the dots is just how many counts each term was counted. So as you can see, there's stress response here, water deprivation, toxin response, and response to salt stress. So the transcriptome is acting how we may expect under stress, and I can move forward with my analyses. A major goal of my project was to see how species respond differently to salt stress. So this is what we're going to do. Before I show you these data, I just want to walk through how we should go about reading this plot because it's kind of a lot of information. On the x-axis is the Arabidopsis C-score. Anything above 0 can be deemed as positively regulated, and anything below 0 is negatively regulated. Same for the Camelina y-axis here. So as you would expect, this quadrant is Arabidopsis upregulated as well as Camelina. This would be Arabidopsis downregulated, but Camelina upregulated, and so on and so forth for the remaining quadrants. So here are all the points. I want to make it extremely clear that each data point is an ortholog between the two species. And by ortholog, I just mean gene that does the same thing. So as you can see, points along this line are behaving similarly, or they have the same expression pattern, but on the other diagonal, we have our genes that are behaving in different patterns. And so I'm interested in data that lie here. But once again, this is still a lot of data. So we wanted to just look at a very specific class of genes that's very important to regulating these genes called transcription factors. Transcription factors bind to the promoter region to turn off or turn on genes. So as you can tell, they're very important. Here are transcription factor families that were very abundant in the data. And these families are involved in stress response and development. This can be read similar to the scatter plot I just showed you a few slides prior. with Arabidopsis on both x-axes. The top figures have our camelina data, and the bottom figures have our Eutrema data. Still, each point represents an ortholog, except between all three species. I'm interested in data that lie in this quadrant, where Eutrema and camelina are both upregulating the transcription factor. However, Arabidopsis is downregulating the transcription factor. And I'm interested in these data specifically because they may be conferring the salt tolerance that we're seeing in Eutrema and camelina, but not in Arabidopsis. However, I don't want to just look at transcription factors because there are other regulators that may be at play. This is an emerging class of regulators called antisense long non-coding RNAs, linked RNAs for short. Antisense long non-coding RNAs are transcribed in the opposite direction of their protein coding gene, and they only regulate the gene that they overlap, unlike transcription factors. In fact, there are about 1,500 antisense linked RNAs that are known to have a protein coding gene in Arabidopsis, which begs the question, are any of these linked RNAs involved in the salt stress response? To begin answering this question, you need to first make sure that your antisense linked RNAs are correlated to your protein coding genes. So next, I'm just going to show you a density curve of correlations, and I will walk you through this data as well. On the x-axis here are our correlation values, and the density axis just tells us how many counts of each correlation we saw. So anything in this box can be deemed a very strong positive correlation above 0.5, and anything in this box can be deemed a very strong negative correlation below negative 0.5. Most known antisense linked RNAs that have been published negatively regulate their protein coding gene, and they would be found in this box here. So I wanted to kind of look into linked RNAs and protein coding genes that may fall here. And we found one called NOG1. NOG1 in Arabidopsis when put into rice. Clearly, I didn't do this study. This is just other, is known to confer drought tolerance. And although I'm studying salt stress, since salt stress and drought stress activate similar pathways and behave in similar manners, this is still a very interesting discovery. In fact, in Eutrema, we identified an antisense linked RNA that is very strongly negatively correlated to Eutrema NOG1. So this is sort of what the gene looks like. The protein coding gene and the antisense linked RNA overlap at their 5' ends, which means the start of transcription overlaps. And here is a visualization of the strong negative correlation. So as you can see, when the protein coding gene is turned on, the antisense linked RNA is turned off. When the antisense linked RNA is turned on, NOG1 is turned off. So this is very interesting data. I got to identify lots of transcription factors in linked RNAs and then kind of narrow down to this specific example that might be used to help identify potential tolerance mechanisms. So overall, I just did the bioinformatics. I didn't really touch any wet lab work. I conducted analyses, and then I identified interesting transcription factors in linked RNAs. And future directions may include identifying more regulators, creating knockouts or maybe overexpression lines, and then evaluating gene expression changes just to make sure that we're seeing on the molecular level what the data suggest to us before. So thank you so much to the Boyce-Thompson Institute. I worked in the Nelson Lab. My mentor, Kyle Paulos, helped me so much, not only with just doing lab work, but with personal and professional development. So I really appreciate that. And shout out to my lab back home, Mark, Nick, and Erica, for giving me the confidence in the scientific community to be able to do what I'm doing here today. I'd also like to point out that a lot of the data was collected at the U of A. And thank you, NSF, for funding me, and Cornell for housing me.
Questions? You did a great job, Sam.
you.
Says Cole too.
Thank you.
And I wanted to know what was like your favorite part of what you did, and then also like your least favorite part. Like what was hardest? Yeah.
The hardest part was the talk. Everything else was pretty smooth. I enjoyed most when Kyle didn't give me the answer to things immediately, when he kind of was like, I'm gonna give you a problem to try and code, like use your resources, use Google, and like look up these documentations to see if you can do it before I help you along. And I really like that. That's my favorite part.
That was real. Thank you.
Oh,
Our question online
No.
is from Mark Bilstein.
oh, yay.
It says, what does NOG1 do in Arabidopsis?
Sorry.
What does NOG1 do in Arabidopsis?
That's a great question. I just know that when put into rice, it confers drought tolerance. But I don't know too much about NOG1 in Arabidopsis specifically. I'm so sorry.
Thank you, Sam, for an amazing presentation. I thought you did an awesome job explaining a bunch of concepts related to biology and bioinformatics. In that vein, I was curious what like spot along the bioinformatics pipeline from start to finish is the most interesting for you?
Bioinformatics pipeline. I feel like there are 4 billion bioinformatics pipelines, but for the bioinformatics I did specifically, I liked all of it equally. I like coding, it's fun. I hope that was a good enough answer.